Dear colleagues, welcome to our dialogue in July. It is almost the end of July, but in spite of vacations, we are here with our dialogues. This initiative of the European Heart Journal has been extremely successful, and we have the pleasure of inviting top leaders in cardiovascular medicine. And during our dialogues, we explore not only their science, but also their human experience, which is inspiring for all of us, and in particular for our younger colleagues. And today, we have the privilege of a famous guest, Professor Lars Wallentin. And the title of the dialogue today is a new look, a fresh look to registries with Lars Wallentin. But you will see, will not just be limited to registries. We will explore many sides of the incredible scientific contribution of Lars Wallentin to cardiovascular medicine. And now it's my pleasure to ask Professor Libby, Executive Editor of the European Heart Journal, to introduce our famous guest. Peter, please.
Well, indeed, Lars hardly needs an introduction, but let me highlight some of the strong points in his resume for those who may not be acquainted with the depth of his leadership and experience. He's a senior professor of cardiology and the founding leader of the Uppsala Clinical Research Center in Uppsala University Hospital in Sweden. And he also, and we'll delve into this in some depth during this dialogue, is the founder of the Sweetheart Cardiovascular Registries, which really has been a model for registries around the world. It's the envy of most registries because of the completeness of the data which are available in a country with a first world healthcare system like Sweden. And he's currently engaged in the development of a conjoint registry that will join all of Europe. This is EuroHeart. So the research group of Professor Wallentin has developed several new concepts. So he's deeply embedded in the randomized clinical trial, but now has quite a bit of interest in the use of registries in order to conduct clinical research in a way to try to shortcut some of the exigencies that are involved in the traditional randomized clinical trial. So we'll have a chance to go into that. And he has wide-ranging interests, as we'll come to in a moment, ranging from antiplatelet therapy to antithrombotic therapy, and also many other aspects of the thread of acute coronary syndromes, which has run through his research career. So he's published widely, more than 800 papers in peer-reviewed international journals. And I think that the list of his research awards would take up the rest of the hour. So I think we'll delve right into the meat of the matter here. Okay. Lars, you started out in lipid metabolism, and then you morphed into thinking about antiplatelet therapy and anticoagulant therapy. So can you give us some idea of what motivated you during the evolution of your research path?
Thanks for a very nice introduction. Where it all started, I mean, it started, well, I don't dare to tell you maybe, but it started in the 1970s. It's quite a long time ago. It's more around 50 years ago. And you need to put this work at that time into perspective. I mean, very little of what we have in cardiology today was really available. Personally, I was working on valvular heart disease, rheumatic heart disease, diagnosing aortic stenosis, mitral stenosis, doing hemodynamic catheterization, sending people to valve surgery, working with the pioneers in heart surgery like Viking Olof Björk and others with the new valves, and also sending the first patient from Sweden to heart transplantation in the early 1980s. So this was really what I was working on, but I needed also do some research, and one of my mentors had a lab that he didn't really like to continue. So I had the opportunity to take over this laboratory, which was focused on phospholipid metabolism and phospholipid. And in some way, I turned this into a lipid lab, and we were doing lipoprotein analysis, of course, but still was focused a bit on phospholipids, working on the lecithin cholesterol acyltransferase mechanism, which was my thesis. But I learned a lot about phospholipids and prostaglandins, of course, and this took us into the area of aspirin and low dose aspirin. So this is where it started, and based on that. Antithrombotic treatment were more exciting when working in the clinical arena, and I was really a clinician for 20 to 30 years, dominantly a clinician, and I like to see some development in the clinical treatments, and then at that time it was really more exciting with developing knowledge on thrombosis as the reason for heart attacks rather than lipids. As you know, it was in the 1980s the thrombolysis treatment was started and the first trials of thrombolytics appeared. And then you realize that maybe also prevention by antithrombotic treatments could be a new avenue. And that's when we started the low-dose aspirin in acute coronary syndrome trial.
Yeah, so aspirin seems to have gotten more and more confusing as the years have gone on. So you're the world's expert here. So maybe you could give us a view of where we stand with aspirin today. Is there any role in primary prevention? And if we say high-risk primary prevention, what do we mean? And I just wonder whether there's an age limit on the use of aspirin in secondary prevention. I just read from the ASPRI study a sub-analysis looking at the elderly with a really very frightening incidence of hemorrhagic problems, including intracerebral hemorrhage. So where do we stand today? What should we do with our patients in primary prevention and also the elderly in secondary prevention?
I think it's less controversial in secondary prevention, where we really started. I mean, in acute MI and acute coronary syndromes, that platelet inhibition is a cornerstone of treatment, and it's still remaining. The question is rather, how many antithrombotic treatment can you tolerate in that setting and for how long? But I think aspirin still is a very effective treatment, and when you add on the P2Y12 inhibitors, maybe you should only for long-term treatment use one of those. And of course, clopidogrel or ticagrelor might be safer on long-term treatment, but until now more costly, but now they are generic. So therefore, we might really see that aspirin is no longer staying for long-term treatment and secondary prevention. In the acute phase, there are really trials showing that if you try to get away from aspirin, there is an increased risk of stent thrombosis. So I think the combination should be used in acute coronary syndromes for at least for a month, maybe up to 3 months. But thereafter, it can be discussed which treatment should be preferred, and the less of an antithrombotic you use, the less bleeding there is, of course. Primary prevention is very controversial, I must say, and I think it should be based on risk maybe rather than age, and we need new tools, I think, to evaluate risk for ischemic events and risk for bleeding events. We have been working a lot on those tools using biomarkers, trying to identify beyond the clinical risk factors, also biomarkers indicating risk for new thrombotic events and also biomarkers related to risk for bleeding. I think this is an exciting area where you need to combine many pieces of information, and there are laboratory data that could be very useful in this setting.
Yeah, so you've not limited yourself to aspirin, which of course is a drug which is well over 100 years old, but you then became very interested in the early days of the low molecular weight heparins And then going back to your interest in valvular heart disease, you've been interested in the direct oral anticoagulants. So how did that journey happen?
I think we started with aspirin, and we looked for some additional possible treatments because we thought that aspirin might not be effective enough to prevent recurrences. And in our very first trial, we used standard heparin, IV heparin. But as soon as we stopped the heparin treatment, there was a rebound. So therefore, we looked for something that could have a more prolonged use. And what was available at that time, which was quite new, was the low molecular weight heparin. So we evaluated that for longer time periods, and it turned out to be, as effective as IV heparin. And at that time, there were waiting times for invasive procedures. People were not going directly to the cath lab if you had a non-ST elevation MI or even unstable angina. You had a waiting time, and during that waiting time, we showed that you could protect the patient by the combination of an anticoagulant, low molecular weight heparin, and aspirin. And we even tried to use low molecular weight heparin for 3 months. But and it turned out to be effective for around 6 weeks, but I think adherence was poor over time, and it was not a practical solution really. So low molecular weight heparin, they are still there for the acute phase as one alternative if you have a longer waiting time. And we know that the combination of an anticoagulant and a platelet inhibitor in the acute setting of an acute coronary syndrome is effective. So this is where it came from, and that also was the reason really why I became involved in other areas like can we do something better than aspirin, and then I became involved in the P2 white valve inhibitor development.
Yes. Yeah, I remember being in Barcelona when you read out the Plato study, which was really a landmark study that changed practice around the world. So, you started...
I can't tell, the experience was interesting. When we started in the 1980s with the aspirin trial, we had really 800 patients or 1,600 patients showing the effectiveness of aspirin with a halving of the event rate, and also low molecular weight heparin, about the same, 1,600 patients in the second trial, showing another halving of the event rates. And then we used an 18,000 patient trial to show the incremental benefit of an P2 white valve inhibitor on top of this combination. So, the cost effectiveness is really deteriorating over time. And I think that's what we see now with all new treatments in the cardiovascular area.
Yeah, we sort of seem to be reaching the limits of antiplatelet and anticoagulant therapy in the ACS arena, where we're balancing, of course, bleeding with anti-ischemic effects. So is that one of the reasons that you morphed to an interest or applying your interest in anticoagulation to atrial fibrillation? So you're now...
the AFib story, that also a little bit came out of the acute coronary syndrome, because as you remember, the first company that developed a new oral anticoagulant was AstraZeneca, and they developed C-melegatran. And we evaluated melegatran in acute coronary syndromes. And it was, of course, equally effective as low molecular weight heparin. It was promising, could be used for long term, and we have seen other direct oral anticoagulants being effective in this setting. And we had designed a trial, a phase 3 trial, but then this development of C-melegatran was stopped because of hepatic toxicity. And that still, when Boehringer Ingelheim was aware of our development, they asked me if I should join the AFib trials, because there were very few cardiologists knowledgeable in the anticoagulant area concerning new drugs. And so therefore I was involved in the phase 2 with the Vigatran, and that took me into the AFib area, and even the involvement with the direct NA inhibitors like apixaban, etc. And this has been a fantastic history. over time, seeing the incredible development and shift in treatment in AFib.
Really fantastic. So let me remind the audience that they're welcome to ask some questions, and we can invite people who are online live to ask questions. But I know that Filippo is eager to continue the conversation. So this is a dialogue. So Filippo, I hand it over to you.
Thank you, Peter. It's a fascinating conversation. And I note that Greek philosophers are always very popular with Plato and our Socratic dialogues. They're always there. But let's come back to science. I've noted that with, as all of us, with many trials, even successful trials, very convincing trials, like the new oral anticoagulants versus warfarin. I mean, messages in trials were extremely consistent and extremely positive, but took some time for this new information to be applied on a daily basis. Do you have the same feeling, Lars? And can we do anything to reduce the lag between new discoveries from trials and implementation of new drugs?
No, I agree. I think we were rather surprised when coming out with the Dabigatran results in 2009, that people didn't immediately accept the result. There was a lot of reluctance, and there were a large number of questions. What about this? What about that? And of course, we didn't have all the answers, but we had a lower rate of strokes. We had lower rate of bleeding. We had an indication of lower mortality, and there was no need for laboratory control, and quality of life of the patients absolutely better. My message was the number needed to treat is 1, because you get rid of the warfarin disease. Because when you treat with warfarin, you get a disease that you need to monitor on a more or less bi-weekly basis. So, but it took definitely a long time, and not until the 10A inhibitor results came out, especially with the apixaban, people became convinced. So it took a number of years, and there were many colleagues that were reluctant because some of them lost their business of providing warfarin care and outpatient clinic visits for these patients, and people cautioned us that if people don't come back to the outpatient clinic on a bi-weekly or monthly basis, we will lose control. But that wasn't the fact, of course. But I think there were many reasons for this reluctance. I think there is, it's a little bit the same with other treatment also in hospital. One way to have a more kind of rapid development, it is really to run the registries as we have been doing, that you can monitor what's ongoing, that you can have an agreement of implementing a new treatment and then monitor the adherence to the new guidelines, etc. And that's what we are trying to do.
Absolutely. Thank you for this information and sharing your views on this important topic. And in the audience, I see Cecilia Linde, President-elect of the European Society of Cardiology from Sweden, but she also speaks excellent Italian, so I live conversation with her in Italian. She's not the president-elect, she is a top scientist and a wonderful person. Cecilia, I'm sure we have questions for Lars.
Yes, I mean, I've been following what you said, and of course, Lars, I know all your accomplishments being a fellow Swede, but I would perhaps like you to talk a little bit about the journey of introducing continuous patient registries as we have in Sweden and what that can accomplish, because that has been a major achievement now, followed by introduction of the EuroHeart.
Yeah, I think the startup of the Swedish registries has become a success story. It is though a long story because it started around 30 years ago by the end of the 1980s. And the story started in one center where we developed a registry on a mainframe computer, and then further development based on the Macintosh, in the Macintosh computer with 512 kilobyte central memory, and where you had all your data on diskettes. So this was the basis for the system at the beginning, and the purpose was really we were concerned about side effects of some treatments, and we like to monitor the side effects. That was the initial story. We like to get rid of lidocaine for ventricular arrhythmias in acute myocardial infarction, because we thought that it's more hazardous than beneficial, and this could be shown. But then we realized that now we also know how people individually treat patients, and there are large variability. And then we implemented this system in 9 centers in Sweden, and thereafter in 18 centers, and finally in 1995, after around 10 years, in almost every center in Sweden. And we had the support by the Board of Health and Welfare, and we had some industrial support also to develop the system into an internet-based system, which was started around the new millennium. And then it kind of was more accepted, and our internet-based system was also a success story, because it was used in several other disease areas. In Sweden nowadays, we have around 100 different national quality registries in different areas of disease. And the Swedish system, starting in acute coronary syndromes, in bypass surgery, in PCI, has also gone into other areas, heart failure, arrhythmia. And the key is really to have an acceptance that physicians or nurses at the centers should enter structured data for every single patient, structured data that can immediately be analyzed online. You can get immediate information now on how you provide care for a specific patient in comparison to other similar patients, or a group of patients this month versus previous treatments. You can monitor treatment over time, and you can compare to guidelines. And then, of course, we bring all of these data together and have databases for the whole of Sweden for epidemiological research. So this has been a great success story, and it has been also a very useful tool for collaboration between all different centers with yearly meetings, etc., and a lot of solidarity to improvement of care. This has been publicly very much discussed in the media, etc., and some resistance, as Filippo told me us before. Some centers did not accept immediately the system, especially as it came from a smaller university hospital at that time. It didn't come out of the big centers. The big centers might have been more reluctant to accept the need of this monitoring than others.
But I
A
think
short
what is
story.
interesting, Lars,
Cecilia, you can update me.
Yeah, I just want to. I remember the days when we said no, they will never accept to enter data. No, they will never accept to compare one hospital to the other, or even worse, compare one doctor to another. But actually, it was done, and it's sort of, you have to face that reluctance, and to for everybody to realize the advantages. And so we have this tradition in Sweden. Other countries do not have this tradition, but human nature and the mentality of hospitals and nurses is probably the same all over the world. So I just can see how great the advantages could be if such a system was implemented more over our Europeans or worldwide countries, in whichever form is feasible in that geography.
Can I ask you, both of you, a question? One of the most fascinating aspects of science is continuity, transition from mentors to trainees who become mentors at a certain point in time. I think that this continuity is the main reason why science develops so quickly, so efficiently. And let's throw some light on Sweden, because Sweden, really, the contribution of Sweden to cardiovascular medicine in the last 50, 70 years has been really remarkable. What about your mentors and your trainees? What about this continuity in your life? Cecilia first, and then Lars.
Well, you know, I haven't been very adventurous. I've been at the Karolinska all my life. Yeah, so I think I had the privilege of having great mentors like you, Lars, like Lars Wallentin, who was the president of the European Society of Cardiology, and also by role models internationally. And that, of course, has to be linked with the personal interest to get to know leading persons in the world and interacting with them and being part of randomized controlled trials. So it goes both ways. I think now it's generally at least encouraged that as much as possible you should do a postgraduate position somewhere else. And of course, if you have that opportunity, that is great, because then you see that you can do things in different ways and you can get a wider contact net. So my philosophy has always been that the doctoral students I have should be better than me, and they should be more creative, more ambitious. And I'm very proud to say that all the heads of cardiology after me are actually my doctoral students. Now it's the third one. So we have a sort of pacemaker dynasty, because I'm a pacemaker scientist from the beginning, starting with Lars Wallentin, myself, Fredrik Adler, Friede Braunschweig, now Marcus Ståhlberg. So I think that how your mentees do is really the best grade of how you perform. And I really believe that you should try and promote those younger than you are, and those that want to come up somewhere in their cardiology career.
I absolutely agree with you. It's really one of the most fascinating aspects of our life, yeah. Academic life. Lars, what about you? Mentors and trainees?
I think, I mean, my majors were more local cardiologists with very skillful in the clinical area and very good in handling patients and colleagues. But in some way I was at a small newly founded university. I had rather to start the research myself, and therefore I'm very much a hands-on person and have developed many things that you have seen over time. And maybe I would have needed some mentoring in certain areas more than I really had at that time. But still, what I'm very proud of that I have mentored a number of people who are succeeding me. So therefore, in all areas where I have been active, I now have successors. And I am really telling them every year or even more often that now I need to resign. And still they ask me to stay all the time. And I take this as a message, maybe I still have something to offer. But as you know, I have several people now in the European Society and Swedish Cardiac Society in different departments. And they are continuing, and they are now reaching, I think, higher levels of science than I ever accomplished. But still, we work a lot together.
Well, I can witness your enthusiasm based on the interesting papers we received at the European Heart Journal from you and your group. Remarkable contributions. But back to Peter. Peter, registries and trials embedded in registries is another aspect, as we have just seen, which characterizes Lars Wallentin contribution to science. Other comments on this important aspect from your side?
Yeah, well,
I think that's a very exciting area, really, because I mean, when we run the registries, these are structured information like a CRF, a mini CRF that is ongoing 24/7. So you have the patient characteristics and you have the key treatments, and in the Swedish system, we also have outcomes, long-term outcomes. We can track every patient's full life based on the personal identifier, and not only survival, because we can merge with other public registries. So we have every readmission with diagnosis, we have every prescription on medication, we have every additional procedure. And this concerns not only the cardiovascular diseases, all types of diseases. We can also go backwards into the healthcare system and get prior history based on prior admissions to hospital. So this gives us very cost effectively a complete information for a clinical trial. And of course, then we realize that, I mean, if we are monitoring patients like this, why not randomize? And that has created a tool. You can do a registry-based randomized trial, and then you don't only randomize to one novel medication versus another, or one new procedure versus another coming from the industry. We can come up with any type of question, and we are very proud of that this registry-based trial was the first to show that oxygen is not needed in the ambulances as a routine for patients with chest pain unless there is a low oxygen tension. So therefore, and this is just a simple question. This treatment has been there for 50 years. You always enter a tube with oxygen. It's not needed, completely not necessary, because it doesn't change outcome. And you can come up, and quite recently, beta blockade was tested versus no beta blockade after acute MI in the routine patients without any sign of heart failure, an ejection fraction above 45, I think. And there was no difference. So some treatment can be eliminated in this way. And we are testing new devices, like the first trial was really on a thrombectomy catheter. Will it be an advantage to remove a blood clot from the infarct-related artery? It looks very appealing. You just remove the clot, you have blood flow. Of course, it should be advantageous, but no, it doesn't matter, because a new blood clot is forming there if you don't put a stent there. And if you put a stent there, anyway, you don't need this thrombectomy device. So all types of questions can be asked in this setting, and you can evaluate both new devices and new medical treatments. And the Swedish system is now oversaturated with these type of trials. I think we are doing around 10 simultaneous trials in the cardiovascular area, but 10 in other areas. And all of these are really high quality trials with enough power and published in New England Journal of Medicine. And usually we have 1 or 2 publications of registry-based clinical trials in New England Journal of Medicine every year. So therefore it is a big success. It's accepted now by FDA. I think on the ESC Congress, the first trial of a completely novel stent, which is tested for the very first time in the registry-based trial in Sweden, and showing whether this new stent technology is advantageous to another. And these trials are also very cost effective. I think the cost is around 10% of a conventional trial. The first trial was maybe only 2% of a conventional trial, but still over time, if you have some FDA requirements on monitoring, etc., it will be more expensive, and also in an international environment. But still, and these trials have now moved into the international area. And of course, this is one of the reasons why we are trying to develop this system for Europe, because Sweden is a small country, 10 million inhabitants. But if we need, let's say, 7,000 patients with acute coronary syndromes in Sweden, it will take us 2.5 years. Not bad for one country though, but still. But if we could involve 10 countries in Europe, we would do it in half a year. And we could come up with many questions that are never asked, never answered, and also collaborate with the pharmaceutical and device companies and offer cost-effective alternatives to the almost prohibitively costly trials that are now needed for approval. So I think this is a very exciting area.
That's really a tremendous advance. And I think that running trials for non-pharmaceuticals or non-branded pharmaceuticals is going to be revolutionary. I see that one of our participants, Ogosa Tabatabe, has a question going back to your early interest in valvular heart disease and anticoagulation. So I don't know, Dr. Tabatabe, do you want to come on and ask your question?
Yes. Hello. Hello. Actually, I wanted to say that warfarin disease, maybe with warfarin disease, maybe a relevant diet, if a relevant diet would be patients, it might make them prevent warfarin disease. Because it's very the nutrition and their diet is very affects this disease very much.
With the warfarin treatment, there are interactions with the diet, of course. Yeah, and they in these trials, I think we tried to improve the warfarin treatment as much as possible when we did the trial. So in order to, regardless of diet, make the warfarin care have as high quality as possible. I don't know if this was your question really, but still, concerning relations between warfarin and diet, we were trying to compensate for that. Still, it's simpler to use the direct oral anticoagulant because then you don't need to care about the diet.
Yes, maybe a simultaneous both instructing the patients with a relevant diet and also a more frequent checkup on the patients and thrombotic status would make this disease, would make prevention with this disease.
Yeah, but in some way, we using the new direct oral anticoagulant, which now are generic and therefore very rather cheap and without any, I think, obstacles to get them, you can care for the patients and have the same protection against stroke as with warfarin, and you don't need to care that much about diet for stroke prevention. That is.
Yeah, the problem is when warfarin works better than these new oral anticoagulants like prostatic valves. But this population where Coumadin warfarin is still needed is a population which is quite limited and perhaps in the future will be even more limited. But Lars, coming back to registries, now with electronic health records, we are going through a new phase. Basically, you started before this was possible, so really more challenging than now. But now all this process of, for instance, trials embedded in registries with electronic health records will become probably easier and more widespread. What are your feelings? Will this facilitate the evolution of what you started many years ago?
I think it definitely will facilitate. It's very exciting to see this development of electronic health records. I mean, the difference between what is recorded in the patient record, it's usually free text. These are not structured variables. You might have a long story about smoking. We just use a question with 3 alternatives: currently smoking, previously smoked, or no smoking. And that's what we are using, and we are directly analyzing. So we like to have structured data and it would need a very advanced artificial intelligence to extract the information from the patient records if the personnel continues to record in the same way as they do now. What we would like to see is that the structured data we are using are integrated as part of the recording in electronic health records. And that's ongoing. I mean, we have several projects in Sweden and several centers doing this. So I think, and we can, for heart failure, we can get, I think, 70% of the variables out of the electronic patient records, but not all of them. So I think a combination of acceptance of using structured data, that is variables, as part of the electronic patient records, then we will have solved the problem, and we don't need, I mean, separate registration. We can have it all integrated in one tool.
Very, very interesting. But in your life, not only trials and registries, but the third passion has been biomarkers. And now I have in front of me two champions of biomarkers, Peter and Lars. Please start a dialogue on biomarkers. Peter, please.
Yes, so you have the opportunity of mining all of your trials, the registries for biomarkers, and you have been able to really lead the development of some novel biomarkers. Now, one in which we share a great deal of interest that I've been working on since before it had the name is interleukin-6. So I know that that's one of your pet biomarkers. I wonder if you could tell us where we stand with IL-6 and then bring us up to date on some of the newer ones that you have covered in your analyses.
Yeah, I think inflammation activity has, I mean, since we started our first trial, we made also these observations concerning inflammatory activity using really whatever marker, and CRP has been the immediately available marker, but it is not the best. And I think IL-6 is definitely more convincing and provides more prognostic information in a chronic situation concerning the risk of cardiovascular events. Unfortunately, there are many different methods to measure IL-6. It is not standardized. I mean, CRP and other indicators are much more standardized. But still, I mean, the strength of IL-6, it is that it seems to be a pathophysiological cause of the disease, part of a pathway that is causal in relation to, as you have shown, Peter, also, and it is even regulation of IL-6 is also genetically associated with cardiovascular disease. So it is like LDL, it is like probably part of a causal pathway for atherosclerosis, and that makes it fascinating. It is not really the strongest marker of outcome in patients with cardiovascular disease, because the markers that we find, if we like to have an estimate of the risk of death or new cardiovascular events or heart failure, markers of underlying clinically apparent or concealed disease, they are stronger prognostic markers, like anti-proBNP, an indicator of heart failure, like troponin, an indicator of myocardial necrosis. They are the strongest markers in almost all settings, both in acute and chronic cardiovascular disease. It is amazing. They are prognostic at different levels, but still, these two are very, very useful markers. They are not causally related to the disease. I think the reason is that they are revealing concealed organ dysfunction or cellular dysfunction in some way. We have been looking into another marker that, as you know, I'm been excited about, which is EDF15. And a very exciting marker, unspecific, related to oxidative stress, related to, I think, cellular aging. It's related to apoptosis, and it is a very strong marker of survival, poor survival, a higher level poor survival in almost all settings, both in cancer disease and in cardiovascular disease. And I am still trying to understand the reason and the mechanism, but probably it's multifactorial. But still, I've seen several cohorts where GDF15 after an acute MI comes out as the strongest prognostic indicator for mortality. It is the same in AFib, same in heart failures. So I think these three markers, and together with the renal function marker, GDF15 reflects the renal function, but otherwise renal function marker. If you are using these four, it is hard to improve prognostication of mortality and new events by adding anything. IL-6 can add a little, but not really clinically useful, but pathophysiologically and useful for decision-making concerning treatments, but not necessarily improving prognostication that much. So this is where we are, and these markers unfortunately always come out because I have been working a lot on these multi-marker assays using thousands of proteins. And we analyze these in our cohorts, very large cohorts, thousands of patients with acute coronary syndromes and chronic cardiovascular disease and AFib. And when the statisticians come back after analyzing, spending lots and lots of money, they come up with, do you like to know, Lars, which is the answer? And they say, NT-proBNP, troponin, GDF15, and possibly IL-6.
Yes, so you've raised...
So and still I have seen all of the others, and we publish, and Filippo accepts some of them for publication, but still it's always the same markers coming up as risk indicators, not as pathophysiological necessarily fact. So this is where we are still there.
Yeah, there are two important lessons which we can extract from what you have just shared with us. First is that you can separate biomarkers into those that are causal and those that are just reporters rather than being causal, but both can be useful if deployed appropriately. And the other is using biomarkers that report orthogonally on different pathways rather than looking, for example, IL-6 and CRP are in the same general pathway, whereas BNP is looking at a completely different biology from the inflammatory biomarkers. GDF15, as you say, is sort of up in the air, but troponin is reporting on injury of myocytes. So if you have orthogonal markers, it's really hard to beat that quartet or a small oligo set. And we certainly do get a lot of papers in European Heart Journal with the multi-markers, and generally the answer is the same that you said. So Filippo, I think you wanted to ask Lars about some of his outside interests as well.
Yes, well, as we say at the beginning, not only the top scientists, but also the human aspects of the life of our guests are important. And let's come to music. Music has been very important in my life. My son is a violin player. Has been important in Peter's life. He can sing arias by heart from several operas. He knows the arias by heart is really culture, is massive, is impressive, not only science. Peter, you are really incredibly encyclopedic. But I understand music has been important also in your life. Lars, can you tell us about?
Yeah, I started to perform as a singer at 5 years of age. I know my mother has told me. So therefore, I have really been enjoying singing in all types of different groups, quartets, and I'm still singing in the choir. So therefore, and I have lost, I think, at least 1 year of my medical education was more or less lost because I was singing too much in different groups in the Uppsala University. I don't regret it at all. On the contrary, and most of my friends are singers and musicians, and as all other singers, I mean, of course, play a little bit of guitar and piano and, you know, things that you need to do. And so I am an eager choir singer. I could be tested on, I think, mainly choir music rather than operas. I enjoy opera, but I prefer singing in smaller groups.
Yeah, I think this is an important lesson. Life is more than science, and the broader is the perspective, even better is the quality of science. Culture is something which is not limited to one aspect, but is something broader. The broader the view, the best the science, perhaps. And Cecilia, what about music? What about the magic of music in your life?
You know, I tend to be an opera person. I am an all-eater. I also like Wagner. So I've been to Bayreuth. I've been to the Ring in Leipzig, in Stockholm several times. And I'm a sort of amateur ballet dancer. So I'm absolutely what we in Sweden call a cultural lady. So if you go to a theater, it's 75% women with white hair. And a little bit the same if you go to the opera. So I think I cannot live without culture, and I'm also a big reader of literature and try to read in the original language: German, French, Italian, English, and even with the limitations, I still think it's fantastic to be able to read in the original language. So I'm a cultural all-eater. Otherwise, I couldn't do my job as a doctor. You need some sort of cultural nourishment to give you the strength and inspiration to continue.
Let me bring up the other for more or less what is necessary, and it is really physical activity in different forms and also being outdoors. I hate physical activity indoors, in gyms. I always am outdoors. I spend my life several hours a day, always in the forest and looking at lakes and enjoying running in forest or even picking mushrooms or enjoying the flowers and the nature from skiing, of course, in winter time and playing tennis in summertime. Golf is too slow for me.
Well, I think that what you are saying is an incredibly important lesson for our younger colleagues. Culture is more than medicine, it's more than science, and produces results in better science and better medicine. But Peter, what about you? Music is important in your life. Your culture is incredibly wide, and I'm sure that also in your case, culture helped your science. What can you tell us?
No, I think it's exactly what Cecilia said and what you've also intimated is that the balance, it uses different neuronal pathways. And it's actually a very interesting subject and one that Peter Sleight really focused on in the latter parts of his career is the relationship between music and cardiology. You know, the science of Weber waves and music and rhythms are very important. It's a topic that we should talk about sometime. But you know, Cecilia, I share your interest in going to the original language because you can merge opera in the original language. For example, Rigoletto is from Le Roi s'amuse, and if you compare the Sardou play Tosca to the Puccini libretto, you understand what a master of the theater Puccini was because it's so dense and concentrated compared to the original, which was a vehicle in Paris in the 19th century. So I think if you go back and read the originals from which the libretti of operas were derived, it's very instructive. I have to say that, you know, my appreciation for Puccini's dramatic sense and how he worked with his librettists and with ricordi was greatly enhanced by reading the original.
I could add one aspect, I think, to this also, why I think we appreciate music and nature, culture, and we could translate that into science and scientific results. Personally, I only believe and like scientific results if they are beautiful, if there is a harmony, if there is a symmetry, if they fit into the pattern. There might be some disharmony, but overall, you should recognize that, look, this is so beautiful, so it is most likely correct, because that's biology. Biology, when we get it right, it's usually beautiful and logical. It is rarely ugly. So, and I also evaluate our own papers, and they should also be beautifully presented and logically presented. And I think that's what makes them big, not only big papers, but big contributions to science. And I think that's an important message. And I like to transfer this to my fellows, etc., that you should look for that. Don't only do the work, do the paper. Look for a beautiful result that contributes to science.
Well, a scientist said some time ago, science is a piece of art, precisely
Absolutely.
what you're saying, and this is true. And what Peter...
Good science is a piece of art.
Science is a piece of art. And what Peter said before about the brain brings me to my beginning of my career, because my first passion was not cardiology, but was experimental psychology. And in particular, I studied hemispheric dominance. And that was mid-70s. And it is when it became clear that our two hemispheres have different functions. The left hemisphere, the majority of people, is the verbal hemisphere, but music and images go to the right hemisphere. So if you feed both hemisphere, one with science and the other with art, the combination is becomes spectacular. It's the physiological evidence of what we are saying. Lars, I have a very last question for you. How can we improve our dialogue? This is a question I ask all our guests. What are your suggestions to improve our dialogues?
Well, I think you have a very nice dialogue. I think the importance is really to bring together exciting people, and you are doing that. And I mean, having you and Peter and you or some other guests having this active dialogue is probably the best concept. But I have no good advice for you. You have worked on this so long. So
the encouragement is to continue, right?
Yes, absolutely.
Well, thank you, Peter. Thank you, Lars. Thank you, Cecilia, for this exciting dialogue spanning from science to art. I remind you that our dialogues are posted in YouTube and X. So if you missed the dialogue anyway, you'll find it in these two platforms. And I'm also happy to announce a next dialogue, which will be something slightly different as we did in the past year. Next dialogue will not be a dialogue like today, but will be the editorial board of the European Society of Cardiology, which will be live streamed from London at the time of the meeting of the European Society of Cardiology. So exceptionally will not be on the last Thursday of the month, but will be on Saturday, 31st at 19. This is an exception. All our dialogues are on Thursday, on the last Thursday of the month at 17. But the next time will coincide with the editorial board of the European Heart Journal. So will be a very interesting occasion during which we will celebrate the success of the European Heart Journal. Thank you very much for your time. Thank you, Peter, Lars, Cecilia. Thank you, all our guests, and we'll stay in touch. Bye-bye.
Thank you. Bye-bye.